Hey guys, Miss King here, and in this screencast we're going to look at DNA replication. Alright, just to go back and review really quickly, this is a strand of DNA, and we know that it's in this sort of twisty shape called the double helix. On both sides of our helix we would have some nucleotides joined together, and then right here in the middle we have our weak hydrogen bonds that hold our two sides of the ladder together. Okay, and that's going to be important when we start looking at the process of replication. Now, replication is a process. That means it's step-by-step series of events that are going to happen. And we have to do replication before our cells can be copied to make new cells. And the DNA, it needs to be an exact copy. It has to be exactly like the original DNA when we make the copies. Cells are going to need to divide in our body. There's lots of things that happen in our body, things like damage. So if you get a cut, you're damaging cells, and we would need to replace or repair those cells by making new cells. Also growth. Anytime you grow and get bigger, you're adding cells to your body. So we would need to make new cells in order to grow. So to make those new cells, we have to make a copy of our DNA first. So we're going to look at the series of events that occurs for replicating our DNA. All right, so for step one, the first thing we need to do is unwind or untwist our DNA. So we're just going to flatten it out so that it looks more like a regular ladder. Step two, we are going to unzip our DNA by breaking the hydrogen bonds here. These are the base pairs, and remember they're held together by very weak hydrogen bonds. So we can literally break those bonds to open up our DNA. And this little area where the DNA strand is coming apart is called the replication fork. So let's make sure we label that. Replication fork. Kind of like a fork in the road. This is a fork in the DNA. All right, for step three, our two new DNA molecules are going to be formed by adding complementary base pairs to each side. So literally, these little base pairs are just going to be floating around in the nucleus, and they're going to come in and find their match, and they're going to pair up and make a new hydrogen bond to connect together. They're going to do this in opposite directions, and that's what we call anti-parallel. So let's say on this side of our DNA, the base pairs are joining the new DNA strand in this direction. So this A will come in, and then the next A will come in, and then the next A will come in, and then another C will come in, and then another G. So they'll basically build the new DNA molecule on this side in this direction. The opposite direction would be to go from, on this side of our DNA, from the top down. So we would go in this direction on this side. So you can see they would go in opposite directions. So that would be anti-parallel. Now, step four, our two new identical DNA molecules are going to be called daughter strands. D-A-U-G-H-T-E-R, strands. Okay, and you can see now we started with one DNA and now we have two. So these are our daughter strands, and they're what we call semi-conservative. What that means is half of the DNA is an original part of the original DNA that we started with, and the other half is brand new, and those were the base pairs that we added during the last step. So over here, this side of this DNA and this side of this DNA would be our original strands, because we could connect those two back together. And then this part and this part would be our new strands. Okay, so half old, half new, that would be semi-conservative. Now, this is a very basic process, a very basic summary of what's going on when DNA is replicated. Please keep in mind that there's a whole lot more involved. There are many more details that we're not going to quite go into, such as enzymes. There's lots of enzymes that assist in this process of making new DNA. Okay, so this is just a basic summary of what's going on during DNA replication.